I'm just going to jump in and welcome you to today's APSA Café, our nearly monthly colloquium featuring cutting edge Anabaptist scholarship. Thank you for joining us. To begin, I'd like to acknowledge Andrea Waxmunsky, an integral member of our scholarship committee here and the one who manages all of this technology to make the event possible. Thanks, Andrea, for all the work you do to make this happen. For those of you joining us for the first time, we just ask that you save your questions for the end of the presentation or put them in the chat, and then our presenter will be able to get to them after the presentation, and we can kind of use those as a jumping off point for our conversation afterwards. Just so you know, the presentation is being recorded, and the first part of the presentation will be available to everyone, and the questions and discussion will be available will be available on a more restricted access. So if you want to be part of the presentation, feel free to join in. That won't be released to the public. And so without further ado, I'd like to introduce today's speaker, Dr. Jonathan Haines. Dr. Haines has many titles. He is the chair of the Department of Population and Quantitative Health Sciences at the School of Medicine at Case Western Reserve University, where he also holds the title of the Mary W. Sheldon Professor of Genomic Sciences, and holds appointments in the Department of Genetics and Genome Sciences and in the Department of Ophthalmology and Visual Sciences. And finally, is also the founder and director of the Cleveland Institute for Computational Biology. So just an amazingly accomplished and internationally recognized researcher and educator. Dr. Haines has extensive experience in all aspects of genetic epidemiology, with a particular focus on illuminating the genetic architecture of complex diseases. So thank you so much for joining us today, Dr. Haines, and speaking with us about the Collaborative Amish Aging and Memory Project.
Well, thank you for that introduction. Let me share screen here. So you should all be able to see that now. There. So I really appreciate the opportunity to talk with you today and to have some conversation. I'm sure that some of this will be, will invoke a number of conversations and questions, and I'm certainly hoping that we will be able to have a good conversation out of this. As you heard, I'm currently I live in Cleveland. I'm at Case Western Reserve University. I did actually, however, start this work when I was actually at Vanderbilt University down in Nashville, Tennessee, some 20-some years ago. There we go. So just a little bit of background about me and why I'm coming at things the way I do. I trained in human genetics, actually genetics and cell biology, technically, at the University of Minnesota. I did a postdoc at Indiana University. My research interests have really focused mostly on the genetic influences, naturally, on neurological disorders, all sorts of different neurological disorders, both cognitive disorders as well as neuromuscular disorders, movement disorders. But my most enduring research activities, which actually started just after I got my degree some long time ago, has been on Alzheimer's disease and dementia. So that's why the focus today is going to be mostly on that. So the first thing that I think I probably should start off with is why would we study genetics? Why should we bother to do this? Almost all human traits are influenced in some way by what we inherit from our parents, by genetics and genetic variation. Some are influenced almost entirely by genetics. Some are influenced very little by genetics, but almost everything has a genetic component to it in some way. So there are 3 billion base pairs of DNA. We shorthand them with 4 letters, A, C, G, and T. Across the 23 chromosomes, you inherit 1 chromosome from each parent. So you end up with 46 chromosomes. That's what the karyotype over here gives you a little idea of what those chromosomes look like. Some of the diseases that we have in humans are caused by genetic variations. So sometimes in a particular location in the DNA, there'd be an A, sometimes there'd be a C, sometimes there might be a G or a T. They can vary a bit. The genetic variations are very rare in the population and may change only one of the 3 billion base pairs, right? So we're looking for a very small thing in a very large pool of DNA. But the reason we want to do all of this, and the reason that so many people work so hard to develop genetic information, is that finding the genetic cause or causes or influences of disease can lead to better prediction, ideally better treatment, and ideally, ultimately, perhaps cures or preventions of some of these diseases once we understand the biology underlying it. So to give you an idea of the scope of the discovery problem that we're dealing with, this is a cell, and virtually every cell in your body has a complete copy of your genome, of the DNA. That's 3 billion base pairs of DNA. What we often are trying to do is to find one or a few of those base pairs that are changed in people that have whatever trait, the disease that you're looking at. And so we're talking 1 out of 3 billion. So it's a really, really small portion of what we're dealing with. Now, to make that a little bit easier to understand the magnitude of the problem, let's talk, if we compare the human cell, the 3 billion base pairs, to a haystack that is 640 cubic yards of hay in it, that's about 8 or 9 feet, about 16 or 17 feet across and about 8 or 9 feet tall. So it's a really big haystack. And if you take a typical sewing needle, which is about 1/100th of a cubic inch in size, stick it in that 640 cubic yards, that's what we're trying to find, okay? So in fact, we really are trying to find a needle in a haystack, okay? And that's actually a pretty big haystack. So this is the magnitude of the problem that we're trying to deal with. Now, we may be wanting to look for several of those needles, maybe even 100 of those needles, but still, it's a really small thing that we're trying to find in this very large pool of length of DNA that we're working with. So it can be very hard to find these genetic variations, and there are several different ways that one can go about trying to find these things. One way is to find these DNA variations is by testing people randomly. Go out and just test a whole bunch of people. This is typically called a cohort study. But in order to find those variations and really know what you've found, you're probably going to have to go out and study tens to hundreds of thousands, maybe millions of individuals. And that's because most of those people aren't going to have what you're looking for. So obviously that's very time consuming and very expensive. Now there's a second way of doing this, is to find individuals that have the trait you're interested in, not just randomly. You go find people that have whatever trait it is, disease it is that you're interested in, and compare them to people who don't have the trait. This is typically called a case control study. The case have the trait, the controls don't. It typically requires fewer people, but you're still talking thousands to tens of thousands of individuals typically. So in order of magnitude, fewer people, but still a lot of people, a lot of time, a lot of expense. Now, a third way, and the way we kind of like, is to find individuals with the trait you're interested in and then look at their relatives, because the relatives are more likely to also have inherited that variation that you're looking for, the multiple variations that you're looking for. This is a family-based study. And if the effect of that variation, that genetic variation is really, really strong, you may only need to do 10 or 15 people to find what's going on. But you might need thousands of individuals if the effects are more complex, more complicated than just a single variation. And that's in part why we've decided quite a while ago that working with the Amish might actually be a really good way of going to try to understand some of the diseases and traits that we're interested in. So as I'm sure most of all of you know, the Amish emigrated from Western Europe in a couple of different waves. There's a religious and cultural isolation from other individuals in the surrounding areas that allows them to maintain their beliefs and their lifestyle. The lifestyle, which is an advantage for us, increases the homogeneity of the environmental exposures. There's less variation of environmental exposures. Formal education typically stops at the 8th grade, but there's some variation around that. There's not very much smoking or alcohol use, which are two very common confounders, if you will, with looking at various diseases. So that reduces the background environmental variation, which allows us to focus more on the genetic variation. The other nice thing working with the Amish is that they tend to have very large civships and marry mostly within their faith. And this leads to large interrelated families. And because of that, we have a lot of genetic relationships that we can work with. And because of the emigration, it's what we call a genetically isolated founder population. Most of the Amish in the United States, and certainly most of the Amish that we are studying, arose or are related to or descended from a relatively small number of individuals. And if those individuals happen to carry one of the generally rare variations, that variation is going to be much more frequent in their descendants in the Amish populations. And if it is, then it should be easier to find. So that's the underlying hypothesis, the underlying reasons why we see some real advantages to working with the Amish. Now, over on the right-hand side over here, we have the areas, the Amish communities that we're working with. Our collaborators who are actually in Miami but study the Indiana Amish work primarily in Elkhart, LaGrange, and Adams counties in Indiana, and we work mostly with the communities in Holmes and surrounding counties in Ohio. So the Collaborative Amish Aging and Memory Project has really been going for almost, or probably a little bit longer than 25 years now. And we've studied various different traits, diseases over that time. So we've studied Alzheimer's disease, we've studied Parkinson's disease, we've studied successful aging, age-related macular degeneration and eye disease, glaucoma, another eye disease, and autism early on. And today, I'm only going to talk about the work that we've done on Alzheimer's disease and age-related macular degeneration, because I think those two compare with each other fairly well. So just a little bit of background on Alzheimer's disease. It affects over 5 million people in the United States now, and that's projected to increase to about 15 million. So it's a huge problem. The prevalence doubles every 5 years after age 60. So by age 85, up to 40 or 50% of individuals may have Alzheimer's disease or at least significant dementia. It's the most common cause of dementia in older adults, and we typically break it apart into what we call early onset and late onset. Those are pretty general terms. Early onset's quite rare. That's having cognitive problems before the age of 60. Late onset is obviously much more common. The clinical diagnosis is correct a lot of the time, but not all of the time. And there's actually growing debate about what's the clinical diagnosis of Alzheimer's disease versus the pathological diagnosis of Alzheimer's disease. That is, after an individual dies, when you do an autopsy and you look at the brain itself, you see this typical, and this is over on the right-hand side, typical collection of what's called amyloid plaques and neurofibrillary tangles. The plaques are outside the cells of the brain. The neurofibrillary tangles are inside some of the cells of the brain. And the brain, of course, looking at the top over here, the normal brain, it looks pretty healthy, and Alzheimer's brain is withered and shrunken as well. So there's a lot going on in the brain, primarily in the brain with Alzheimer's disease. We do know that genetics is very important in Alzheimer's disease. Obviously, I wouldn't be studying it if we didn't think so, but environmental factors are also implicated. Now, one of the reasons that we really want to understand the genetics and therefore ultimately understand the biology of Alzheimer's disease is because there are very limited treatment options. The treatments that are out there today only treat the symptoms, not the causes. They don't really stop the progression of the disease. And virtually all the drug trials that have been done have failed. There's a lot of controversy about that, but virtually all the drug trials have failed. So we still need to try to figure out how we can get better treatments for this. Now, I'm going to give you just a brief idea of the timeline for genetic discoveries in Alzheimer's disease. And for right now, I'm just going to focus on this early part, this gene called ApoE, apolipoprotein E, that we discovered back in 1993, so quite a while ago. That is very, very important in Alzheimer's disease. We'll talk about the other stuff a little bit later, but for right now, that's important relative to our work with the Amish. So let me tell you a little bit about ApoE. ApoE is a gene. Everybody's got the gene, but there are three common alleles or three common variations that are in the population. There's what fairly simply we call ApoE2, ApoE3, and ApoE4. The ApoE3 allele or variation is the one that's by far the most common in the population. Most everybody has a copy of the ApoE3 allele, but there's the 2 allele and the 4 allele as well. The ApoE4 allele is the one that we discovered back in 1993 that carries a very, very significant risk, increased risk for Alzheimer's disease. So if you have one copy of that, your risk is 3 times what it would be if you didn't. And if you have 2 copies, the risk really is pretty high. It's up to 12-fold increased risk. So ApoE4 may explain a fair amount of all of Alzheimer's disease. On the other hand, the ApoE2, so you can get the other side of this, the ApoE2 variation carries a protection against Alzheimer's disease. So if you've got a copy of the 2 allele, your risk is actually reduced relative to the general population. So that's really, it was really quite interesting to see that you could get either protection or risk in the same gene. And as I said, the ApoE3 allele, which is the most common, is pretty neutral to risk. So I'm not going to go through, so that's what we did back in 1993. I'm not going to go through all the history. I'm not going to explain what the dark years were or anything like that. I just wanted to point out that we're going to move all the way to 2019 in terms of the progress in Alzheimer's disease. And this is pretty much where we are in what we've been able to explain. And we don't have to worry so much about what all these other things are, except for the big green blob over here, which means there's still about half of the genetic influence in Alzheimer's disease that we do not know. And we still need to try to find that and understand where that is. And that's where our work with the Amish starts to come in. So why would we study Alzheimer's disease in the Amish? Back in the mid-1990s, there was an epidemiologic paper that looked for cognitive impairment and identified that it seemed to be somewhat lower in the Amish than in the general population. And because we knew that work was ongoing, we were actually able to look at the Adams County Amish a little bit earlier than that and discovered that the ApoE4 allele, the one that carries all this big risk, basically isn't there in the Adams County Amish. So they're all 3 3s and have copies of the 3 allele. Some of them have copies of the 2 allele. But the cognitive impairment did seem to cluster within families, so that suggested maybe there's a different genetic effect going on in some of the Amish. So we decided that that would be a fruitful research to be doing in the Amish. So how are we going about doing this? How have we been for the last 25 years? We can't use the usual methods of ascertainment of enrollment that we would do in the general population. We're not going to the clinics and things like that. A lot of that's because the modern communication, although that is shifting some now, we can't call them up on the phone for the most part. internet, things like that. So contact is we've generally been doing this door to door or by mail. We have a number of ascertainment personnel, and I've seen some of them are on today, who live in the local area, in and around Holmes County in our case, or Indiana in that case. And so they know the communities, they know the area really, really well. And so it's much more effective for us to have those individuals go out and contact the Amish communities, the Amish families, and work to enroll them in our studies. And we've actually done quite well with that. We think that we've enrolled approximately a third of the Amish population over the age of 65 for one or more of our studies. And obviously that shifts over time over the last 25 years. And we've done some things like in Adams County, we've actually been able to ascertain most of the folks over the age of 85. That was for a successful aging study that I'm not going to talk about. And one of the things that's just been delightful for us and so wonderful has been the overall participation rate is extremely high. The Amish individuals and the Amish communities have been very welcoming to us and very supportive in the work that we've done. So where are we with a lot of that work? We've enrolled over 2,500 individuals across those different studies. Almost everyone who we've enrolled, and we have focused on individuals that have tended to be older, so old over the age of 50, and most of them over the age of 65. Almost everyone has had some level of cognitive testing done. Almost everyone has had some level of genotyping, of being able to look at some portion of their DNA. So we are obviously collecting blood samples when we talk to these folks, as well as doing the cognitive testing. And over 1,100 of them have had what we call whole genome sequencing, where we actually have attempted to get a look at all approximately 3 billion base pairs. There's a lot of detail in there, but we've tried to look at that. So there's a subset of individuals where we have a huge amount of genetic information that we can start to work with. Those data are really quite recent. So one of the other nice things that for a geneticist is that, of course, the Amish are very interested in their genealogy and their descendants and their ancestors. And so we have, with a lot of work by a lot of our, the grad students, the postdocs, and particularly the ascertainment personnel, to be able to connect all of those individuals, those 2,500 or so individuals, who are all the ones down here in these bottom two generations, into a single, incredibly complex, you can see all the lines everywhere, incredibly complex 15 generation pedigree. So all the relationships that we know about and all the complications can actually be drawn out like this. It's an amazing thing to look at these things. Obviously, completely useless for trying to do analysis, but it's a really cool picture. So there are actually two sides to Alzheimer's disease that we are looking at. Most of the studies, and this is true across the field, across the world, most of the studies have tested for genetic variation that increases the risk of developing Alzheimer's disease. It's a logical thing to do. The goal is to find what causes Alzheimer's disease so that you can then try to figure out how to slow or stop it, prevent that from happening. But there's another aspect to this, and many fewer studies have tested for genetic variation that seems to prevent or protect individuals from developing Alzheimer's disease. This would be like the ApoE2 variation allele that does seem to give some protection. Well, maybe there are more than that. So the goal there is to identify the protective effects and try to understand how it's protective and copy that or amplify that protective effect to prevent the process from happening. Now, the nice thing with our work with the Amish is that we can do both of these things in a single study. And I'm not going to go through all the details about how we're doing that. I'm just going to talk a little bit about some of the results that we have, and then we can open it up for all sorts of discussion. So what have we discovered so far with Alzheimer's disease? we know that what is underlying Alzheimer's disease in the Amish is somewhat different than in the general population. And that's what this diagram on the left-hand side is trying to show. If you look at this genetic risk score, which is really just a summation, an overall prediction of how much genetic risk anybody has, it's actually much lower in the Amish individuals that have dementia than it is in the general population in the unrelated individuals. And so that suggests that it's not what we know, which has mostly come from this group, is not explaining as much of Alzheimer's disease in the Amish, which means there must be something else going on. Now, that's very general. Where might these genetic variations be? Can we find them in some of those chromosomes, try to narrow it down to those individual variations? And we're not there yet, but we do have a couple of regions on several different chromosomes where we do think that those variations might lie. There's one on chromosome 2, chromosome 3, 9, and chromosome 18. So there is, we have some evidence for where some of these are, and what we're trying to do now is go in and try to sort through and see if we can find them specifically. Now, what have we discovered so far with cognitive preservation? So the other side of things, this is the newest work that we're doing. And where those genetic variations might be, we do have some evidence that there's something going on chromosome 12 in these regions. And you don't have to worry about all the details here, but these high peaks mean that there's probably something going on. So, in this region of chromosome 12. And then taking a very, very different approach to how we're doing this. So this is transmission. I should say the chromosome 12 is transmission of cognitive preservation in families. So who's related to who, and have individuals that have really good cognitive preservation, do they share the same part of a chromosome, and in this case, chromosome 12. This one actually looks a little bit differently, and it's looking at individuals, at what age are they still cognitively normal, cognitively unimpaired? And when you look at that, we can actually identify a couple of additional places where we think there might be one of these genes. And in this particular case, the one we're most interested in is this SHISA6 gene that we're trying to start to look at in more detail. So I'm going to turn just for a little bit and talk about age-related macular degeneration, which is another thing that we've been studying in the Amish. Basically, what this is the macula is at the back of the eye. It's part of the retina. It's what gives you your very clear central vision. And obviously, degeneration. The macula is degenerating. It's dying, and so the vision isn't nearly as good. And eventually, if it's really severe, you lose almost all vision. We know for AMD, or age-related macular degeneration, that there are strong environmental risk factors. Age, obviously, smoking is a big one, a high-fat diet is another, and then hypertension is another one. But we also know that genetics is really important. And I'm not, again, going to go through all the details of how we got here, but we did identify a few years ago, not in the Amish, but in other groups, that there were 52 different regions that we think that influence the genetics, influence the risk for macular degeneration. And so we used that information that we generated a few years ago, again, to look to see whether those same variations were representative were influencing macular degeneration in the Amish. And just like with the Alzheimer's disease, the answer is it's not the same set of genetic variations. And again, if you look at the Amish individuals with AMD, they have a much lower overall genetic score than the non-Amish cases, and that suggests that what we know, the genetics that we know from the general population, is not having the same influence on the Amish. So one of the other things because we have these families, maybe we can look to see if there are rare variants in some of the Amish. And so there were two very common variants, not rare, that we knew about in the general population, this Y402H and A69S. You don't need to know the details of that, but it's in a gene called CFH and a gene called ARMS2. And we had a nuclear family in the Amish where 4 individuals had macular degeneration, but they didn't have any of these common variations. And so we thought, well, maybe they've got something different. And in fact, we looked within these genes and we found that there was a different variation. Again, we specifically call it P503A, that's in the CFH gene that these individuals shared, and might, we thought, well, maybe that's what's causing or influencing macular degeneration in these individuals. Now, since they were in the Amish, we said, well, there are a lot of other related, they have a lot of brothers and sisters and cousins and second cousins and third cousins. And we thought maybe we should look among them to see if there are more of these P503A individuals. This is work that actually Andrea has done. And we found 52 individuals that carry this variation. What's really interesting is this variation is almost doesn't exist in the rest of the world. So there are very, very few other people in the rest of the world that have this. In fact, we know of maybe 10 or 20, and there are 52 right here. And most of these are actually in the Holmes County Amish. So you can actually identify all of those individuals and how they're related to each other. That's what this is over here with these goldish individuals. And what we've been able to do in looking at this, and again, this is a lot of work that Andrea and others in the lab have done, is that we know that the P503A variation is associated with developing AMD in the Amish. So we tried to understand a little bit about the biology. Well, why is this case? So we looked at some very simple, not simple, but the sort of the most obvious things to look at. One is, does it change how that gene is expressed, how that gene is turned into proteins? Doesn't seem like that's the case. We looked at the actual protein expression. Doesn't seem to be that either. But there's a possibility that it is actually influencing levels of other proteins that are being expressed in the blood samples. So there's a lot more that needs to be done with that, but it's a really, really interesting finding. So I'm gonna end there and open it up for conversation. I just wanted to acknowledge there's a huge number of individuals who have been involved at one point or another here at Case Western Reserve, also at Vanderbilt when I was still down there, at university hospitals here in Cleveland. We have collaborations on macular degeneration with the University of Pennsylvania, who's looking at the Lancaster Amish. I didn't talk about that. And we have some collaborations as well with some folks out in California. And then our close collaborators at the University of Miami that are the ones that work with the Indiana Amish. And a lot of that genealogical data is stored in the Anabaptist Genealogy Database, the AGDB, that is at NIH. And so we've worked with all of these folks. It's a great collaboration. It's been one of the most, probably one of the most enjoyable collaborations that I've had. And of course, we need to acknowledge and thank all the Amish individuals and families that have participated and continue to participate in our studies. So I'm going to stop sharing now and open it up, I guess, to questions, conversations, whatever folks want.